Hello everyone. So we shall discuss today about the continuation of the Gaucher disease, and then we'll go to Niemann-Pick disease. So Gaucher disease, we all know there is accumulation of the Gaucher cells, which are nothing but the lipid-laden macrophages. How will you detect them? In bone marrow biopsy. When you will do the bone marrow biopsy, you will detect the Gaucher cells presence. That will appear like a crumpled tissue paper. Gold standard in Gaucher disease, the gold standard detection, gold standard diagnosis for Gaucher disease is just enzyme assay. Enzyme assay is the gold standard, but not gene sequencing. Okay, gene sequencing is not the gold standard for Gaucher. The enzyme assay, that is the activity of the enzyme beta glucocerebrosidase enzyme, the activity of the enzyme, the detection of the activity of the enzyme is the gold standard test in Gaucher disease. For enzyme assay, always remember the cells which are chosen are WBC or the fibroblast present in our skin. स्किन से फाइब्रोब्लास्ट लेते हैं, in that cell or in the WBC we do the enzyme assay, and this confirms the Gaucher disease because the enzyme activity will be less than 15% of what is desired. If the enzyme activity is less than 15% of what is desired, this confirms the deficiency state of beta glucocerebrosidase. So if I ask you what is the gene defective, we have to tell that it is glucocerebrosidase gene defective, GBA gene, that results into deficiency of the enzyme beta glucocerebrosidase enzyme, which result in accumulation of the glucocerebroside accumulation, and this condition is common. This condition of Gaucher disease is common in Ashkenazi Jews. So Jews, you can remember in short, just in Jewish people, this disease is more common. Ashkenazi Jews. Let me tell you about something about cherry red spot that will confuse you. Okay, cherry red spot. Okay, so suppose this is the phobia. Okay, at the center of the phobia, which is more pale, you will have a red spot. So this thing is your phobia. This condition is found in 4 of the sphingolipid store disorder, and of them Gaucher is not one. So it is not found in Gaucher and not Fabry. So Fabry and Gaucher ko chhod ke, other than these two, the remaining 4 conditions that we have in the sphingolipid store disorders, they have this condition, cherry red spots. Obviously your Niemann-Pick disease, specifically your Niemann-Pick disease type A. Niemann-Pick disease has 3 types, type A, type B and type C. Type A and B, they are both due to deficiency of the enzyme acid sphingomyelinase. It is in short written as ASM, acid sphingomyelinase. We already know that it is sphingomyelinase, but we now come to know it is acid sphingomyelinase. And type C is not due to any enzyme deficiency like your type A and type B. Type A and type B is due to enzyme deficiency. Type A and B is due to enzyme deficiency. Type C is due to deficiency of the gene which is responsible for the production of proteins which are responsible for the carriage of the unesterified cholesterol. Suppose this is a lysosome. Inside the lysosome, this is a lysosome. Inside the lysosome, this is a box which is nothing but your unesterified cholesterol. This unesterified cholesterol has to be transported out of the lysosome. For this purpose, one carrier protein will carry it towards this transporter protein, and then this transporter protein will pump it out. The carrier protein which carries this unesterified cholesterol towards this membrane protein which will pump it out, this protein which is carrier protein is produced by the gene NPC2, and this transporter protein is produced by NPC1 gene. Mostly in the Niemann-Pick disease type C, the gene defective is NPC1, that is the gene producing the transporter protein. This is 95% of the time mutated. 5% of the time this can be due to NPC2 gene mutation. So I hope you are clear about the Niemann-Pick disease type C, type A and type B. Type A and type B, they are due to this enzyme deficiency. Type C is due to this gene defective, which is responsible for the defective intracellular cholesterol trafficking. All right, D is capital. So the condition is defective intracellular cholesterol trafficking. Unesterified cholesterol is not able to be pumped out of the lysosome. So in Gaucher and Fabry disease, which is X-linked Fabry, Gaucher and Fabry, we don't see cherry red spot. But in Niemann-Pick disease, specifically Niemann-Pick disease type A, why? Because Niemann-Pick disease type A and type B, what is the difference between the two? Although they are due to the same enzyme deficiency. Niemann-Pick disease type A is the neuronal form. Type B is not neuronal form, it is just visceral form. Type A is known as neurovisceral. Type B is only visceral. So because it is a neurovisceral form, there is a neuro involvement, and that is why the child will generally die before the age of 2 to 3 years of age. But in visceral form, the child will live up to adulthood. Up to adulthood, the child will survive. In the neuro form, we have the cherry red spot. Okay. Then your other conditions, Tay-Sachs disease. Now you will tell me what is enzyme defective in the Tay-Sachs disease. We know it is hexosaminidase A. Niemann-Pick disease type A and type B is due to deficiency of the acid sphingomyelinase, and then Tay-Sachs disease is due to deficiency of the enzyme hexosaminidase A, which will lead to accumulation of GM2 ganglioside. We know what is M and we know what is 2, right? It is the salic acid residue in the ganglioside. How many salic acid residue is there? One salic acid residue in the number 2 position. So GM2 ganglioside accumulation in the Tay-Sachs disease. And the enzyme we have studied hexosaminidase A. We haven't studied about Tay-Sachs disease in detail. We will study the Tay-Sachs disease in detail. But coming to the cherry red spots, we have Niemann-Pick disease type A and Tay-Sachs disease, and the other two sphingolipid lipid storage disorders we have studied is Krabbe and metachromatic leukodystrophy. These two are left, Krabbe and metachromatic leukodystrophy. Of these two, in metachromatic leukodystrophy, you will find cherry red spot, and cherry red spot is not found in Krabbe disease. तो थोड़ा सा मैंने confusion करके बोल दिया था. So it is not found in 3 conditions: Gaucher, Fabry and Krabbe. In Gaucher, Fabry and Krabbe, you will not find the cherry red spot. You will find that in Niemann-Pick disease type A. You will find that in Tay-Sachs disease, and you will find that in metachromatic leukodystrophy. So Niemann-Pick disease type A, Tay-Sachs disease, and lastly metachromatic leukodystrophy, which is due to the enzyme defective is aryl sulfatase. In these 3 conditions, you will find cherry red spot. But there is 2 more sphingolipid storage disorder which I haven't told you because it would have become much lengthier. But 2 more conditions are there. One is GM1 gangliosidosis, Sandhoff disease. Like you have GM2 gangliosidosis. You can say Tay-Sachs disease, because in Tay-Sachs disease there is accumulation of the GM2 ganglioside. So Tay-Sachs disease can be sometimes called GM2 gangliosidosis. But GM1 gangliosidosis doesn't have any such name, so it is just called GM1 gangliosidosis. So in GM1 gangliosidosis and Sandhoff disease, these 2 are also your sphingolipid storage disorders. GM1 gangliosidosis and Sandhoff disease. In these 2 conditions also you will find cherry red spot. So there is a similarity between Sandhoff disease and Tay-Sachs disease. In Tay-Sachs disease, it was deficiency of the enzyme, you recall, hexosaminidase A. But in Sandhoff disease, there is defective of both hexosaminidase hexosaminidase A and B. So both hexosaminidase A and hexosaminidase B is defective in the Sandhoff disease, and as a result, just like Tay-Sachs disease, there is also accumulation of GM2 ganglioside. GM2 ganglioside. So both Sandhoff disease and Tay-Sachs disease results in accumulation of the GM2 ganglioside. In Tay-Sachs disease, it is just hexosaminidase A. In Sandhoff, it is both A and B. In GM1 gangliosidosis, there is accumulation of the GM1 ganglioside, which is due to the defective enzyme beta galactosidase. If you remember, there is one condition, there is defective alpha galactosidase. Which is alpha galactosidase? It is Fabry. In Fabry, the defect is alpha galactosidase. In GM1 gangliosidosis, it is beta galactosidase. So if I ask you, what is the defect in the Gaucher disease? You have to tell me it is beta gluco. It is beta glucocerebrosidase. In Niemann-Pick disease, you tell me what is enzyme defective? It is acid sphingomyelinase type A and type B. Type C, it is NPC1, NPC2. In Tay-Sachs disease, it is just hexosaminidase A. In Sandhoff, it is both hexosaminidase A and B. In Fabry, it is alpha galactosidase. In GM1 gangliosidosis, it is beta galactosidase. Okay. Finally, we have metachromatic leukodystrophy due to aryl sulfatase A. Aryl sulfatase A. Remember this. A. And lastly, Krabbe. Krabbe disease is due to GALC. What is the full form? Galactocerebrosidase. GAL. Galactocerebrosidase. Accumulation of galactocerebroside. Krabbe's disease. This is also very lethal, like your Gaucher disease type 2. Type 2 is lethal. Niemann-Pick disease type A is lethal within 2 years of age. Type A also dies before 2 years of age. Gaucher disease type 2 dies before 2 years of age. Niemann-Pick disease type A dies before 2 years of age. Krabbe's disease also dies before 2 years of age. Other name for Krabbe disease is globoid cell leukodystrophy. Other name for Krabbe, globoid cell leukodystrophy. Also in Tay-Sachs disease, the child will die between 2 to 4 years of age. So Niemann-Pick disease type A, Gaucher disease type 2, and Krabbe, they'll die within 2 years of age. Also Tay-Sachs disease is also similarly lethal. The child will die within 2 to 4 years of age. Sandhoff disease has two forms. Sandhoff disease has two forms, infantile form and the juvenile form. Infantile form is severe. This will die again by 2 to 4 years of age. Most child do not survive 2 to 4 years of age. The juvenile form is a slow progression disease, and in this the child may lead up to adulthood. So Sandhoff. Acha, the gene which is defective in Sandhoff, okay, the gene which is defective in the Tay-Sachs disease is known as hex A, which code for only hexosaminidase A. But in Sandhoff the gene is hex B, which codes for both A as well as B. Okay, so hex A versus hex B. Hex B is both. Now let me tell you some important information which is MCQ related. So we know about Gaucher disease, right? You You can remember that the gene that was defective was GBA gene, that is present in chromosome number 1. For the Niemann-Pick disease, which is type A and type B, we have the gene SMPD1 gene that codes for acid sphingomyelinase. Okay, SMPD1 gene codes for acid sphingomyelinase, and this is present in chromosome number 11. Gaucher GBA gene, Niemann-Pick disease type AB, SMPD1 gene. SMPD1 gene is responsible for production of acid sphingomyelinase, and this is in chromosome number 11. Okay, then Niemann-Pick disease type C. This is A and B. Niemann-Pick disease type C. This is NPC1 and NPC2. Okay, this is cholesterol transport defect. Okay, now move to Tay-Sachs disease. We have already studied the Tay-Sachs disease is due to deficiency of the gene, defect of the gene HexA. This is present in chromosome 15. Sandhoff disease is due to hex B, which is present in chromosome number 5. I don't like to teach like this, but you have to remember. So Gaucher disease is type 1, it's the chromosome number 1, which codes for the gene GBA gene. NPD type A and type B, we know the gene SMPD1 gene, chromosome 11. NPD, Niemann-Pick disease type C, it is the defective intracellular cholesterol transport, NPC1, NPC2. Tay-Sachs, hex A, Sandhoff, hex B. Gene, you have to remember. Chromosome, you may not remember, but remember at least Gaucher, chromosome number 1. NPD, second most important, Niemann-Pick disease, chromosome 11. Tay-Sachs and Sandhoff, hex A, hex B. Tay-Sachs, chromosome 15, Sandhoff, chromosome number 5. Let's continue with this order. So next, Fabry disease. Fabry disease is due to the enzyme alpha galactosidase, right? Alpha galactosidase, whereas beta galactosidase deficiency was GM1 gangliosidosis. In GM1 gangliosidosis, it was beta galactosidase. In Fabry, it was alpha galactosidase. This Fabry disease, which is due to alpha galactosidase deficiency. Fabry disease is chromosome X. We know it's X-linked disease, so it is chromosome X, and you can remember the gene. It is GLA, galactosidase alpha. Remember this: A for alpha, GL for galactosidase. So GLA, chromosome X. And GM1 gangliosidosis, it is GLB, B for beta. So GL yani ki galactosidase. GL means galactosidase. A for alpha, B for beta. So you can easily remember, if you can remember this, that Fabry is alpha, GM1 gangliosidosis is beta. So GLA, GLB, both are galactosidase deficiency. Obviously Fabry will be in chromosome X, and G1 gangliosidosis is in chromosome 3. Then metachromatic leukodystrophy, and the last one, Krabbe, MLD and Krabbe. MLD is due to aryl sulfatase A, AA, aryl sulfatase A. Is in short written as the gene is written as aryl sulfatase A. Aryl sulfatase A. Krabbe's disease, galactocerebrosidase. Remember, it is galactocerebrosidase. So it is galactocerebrosidase, GALC. Aryl sulfatase A, galactocerebrosidase. Aryl sulfatase A, ARSA, galactocerebrosidase, GALC. So MLD is aryl sulfatase A, ARSA. Krabbe is galactocerebrosidase, so GALC. Lastly, have another sphingolipid storage disorder, and that is Farber disease. Okay, Farber disease is due to the defective enzyme ceramidase, and this gene is known as ASAH1, ASHA gene. Farber disease, which is also known as Farber lipogranulomatosis, lipogranulomatosis. Okay, Farber lipogranulomatosis. It is due to the enzyme which is responsible for coding acid ceramidase. Acid ceramidase. It is written as ASAH1, ASHA1. This Farber disease is in chromosome number 8. So let me ask you questions. Which gene is defective in the Tay-Sachs disease? Which gene is defective in the Tay-Sachs disease? Answer is hex A. Tay-Sachs disease, hex A. Sandhoff, hex B. Hex A, chromosome number 15. Hex B, chromosome number 5. Hex A, only hexosaminidase A. Hex B, it is defective hexosaminidase A as well as B. This is Tay-Sachs disease. This is Sandhoff. Okay. The next question asks you. GBA gene mutation. GBA gene mutation will result in which disease? Very simple. GBA gene, it is the glucocerebrosidase, that is your Gaucher, that is your chromosome number 1. Okay, now GLA gene disease, mutation disease, GLA. Remember, GLA is your Fabry, because A for alpha, GL for galactosidase. So alpha galactosidase, GLB is beta galactosidase. Beta galactosidase, GM1 gangliosidosis. Alpha galactosidase, Fabry's disease. GLA gene, Fabry disease, chromosome X. Whereas GLB is present in chromosome number 3. So beta galactosidase, which is defective in GM1 gangliosidosis, this is present in chromosome number 3. Next question. Which gene is associated with Krabbe? Krabbe's disease, which is globoid cell leukodystrophy. Krabbe disease is due to deficiency of which defective gene? It is, we know it is galactocerebrosidase, so write it galactocerebrosidase, GALC, and this is chromosome number 14. So Krabbe is GALC, chromosome 14. Niemann-Pick disease, we know Niemann-Pick disease type 1, type A, B, C, sorry. So Niemann-Pick disease type A and type B, this is due to the defective enzyme acid sphingomyelinase, and Niemann-Pick disease type C is due to the defective enzyme enzyme gene NPC1 and NPC2. The gene is SMPD1. This is present in chromosome number 11. For this, it is very difficult. NPC1, NPC2. NPC1 is present in chromosome number 18, which is the most commonest defect 95% of the time, and NPC2 is present in chromosome number 14. तो धीरे धीरे हम लोग याद कर रहे हैं, एक बार में बताता तो मैं ज़्यादा हो जाता. NPC1 is chromosome number 18, NPC2 is chromosome number 14. Which gene is defective in metachromatic leukodystrophy? The gene and the enzyme. We know the enzyme, it is aryl sulfatase A, metachromatic leukodystrophy. So aryl sulfatase A, and the chromosome is chromosome number 22. Okay, chromosome number 22. And Farber disease, which is known as Farber lipogranulomatosis, which is due to the enzyme defective. Tell me the enzyme. Farber disease. Yes, ceramidase, and the gene is ASAH1, acid sphingomyelinase like, that is the full form of ASHA, ASAH, sorry. ASHA, ASH, ASHA, acid sphingomyelin like, acid sphingomyelin like, but the enzyme is ceramidase. Farber or Farber lipogranulomatosis, ceramidase deficiency, ASHA1 gene present in chromosome number 8.